So what we work on is something called genetic control, which is an exciting technique and has a lot of potential because it's a way of introducing genetic traits into a population. In our case, we work on mosquitoes, the mosquitoes that transmit malaria. And the ultimate way of blocking malaria transmission is to actually control the mosquito. Things like insecticides have worked very well, but they have their challenges, one of them being resistance and the cost and the overall logistics of implementation. Whereas with genetic control, you actually release mosquitoes that have a modified trait and you try and spread that into the population. It takes some quite complex molecular biology and genetics just to understand it, never mind to actually build it. And so that's why we are trying to build that capacity within these African scientists such that they can become leaders and inform around these technologies and allow those countries to make more informed choices about whether to adopt this type of technology, as well as having the capacity internally to actually develop and lead in it.
Mosquitoes need to take the blood for the eggs maturation, and by taking this blood from human, they can transmit the parasite to the human and make it ill. So we are investigating the potential genes that are involved in that process in order to develop genetic control tools. We look for the specific gene and then break this gene function in order to impair the mosquito to be able to use this gene for the blood digestion. So, we did a knockout of the gene. That means we silence definitively the gene by CRISPR-Cas9 specific tools, which is a high precise genetic engineering tools.
We've had these breakthroughs before in our lab, but this is the first time that the Burkina team, anyone from Burkina actually, has made transgenic mosquito, so it is a monumentous milestone. So the idea is that with these skills that they've now picked up, the molecular biology and genetics, we're at the same time in collaboration with IRSS in Burkina Faso building that molecular lab capacity and insectary capacity, as well as the methods to inject the mosquitoes within that country itself, so that they should be eventually equipped to use the skills they've picked up here and work in those labs there and transfer that capacity across to Burkina.
To come here in LSTM is a really opportunity for us because we learn a lot of things with Tony Nolan group. Thanks for Tony for all he has done, because we learn really a new tools, and we are confident and we can maybe carry these tools that we learn here in Burkina Faso.